So what we're going to examine now is what a typical neuron looks like. This is what the brain and spinal cord is made out of. It has a bunch of these completely laid out and bundled together in the millions to carry out all the functions we need to do. And so to start, we have three regions to consider. We have our dendrites, which are going to be these shorter extensions. These shorter extensions are going to receive action potentials. They're going to receive nerve impulses from other neurons in a manner like this. Like when you see a connection, you don't have to flip pages, I'll do it for you. But you can see here a neuron is connected to another one. And so it'll relay a message, and you can see that it connects at the side with the dendrites. And so they exhibit some polarity, or said even easier, they exhibit different sides. Side like this side and this side down here. Where the dendrites, their main job is to receive stimuli. So important, we highlighted twice. Dendrites, they are highly branched, and that's where the CNS neurons receive most information. The next structure is the cell body, and that cell body is like any other cell, just the container of all the contents inside. That's going to be the cell body, where it has a plasma membrane like any other cell, has a nucleus like any other cell, has all the organelles that any typical cell has, but it has them in a unique arrangement to make this a new cell. Remember that all cells have all those organelles you learned about in chapter 2, but they have them in unique combinations to do very unique things. So with that in mind, the cell body has some terms around it. It's going to contain a cytoplasm. In this chapter, we don't call it cytoplasm, we call it perikaryon. This word is the same word as cytoplasm. Peri in Latin means around. Keri comes to mean in Latin nucleus, so around the nucleus, all the area around the nucleus, which is the cytoplasm, and it contains all those organelles. And one of the most relevant things about it today is that it synthesizes neurotransmitters. And neurotransmitters, at the end of the day, what they are is that they are hormones sent between the brains. And I wrote this somewhere else for you on the packet. You don't have to write it down. But you'll see synthesizes neurotransmitters, which are hormones of the brain. Said simple. Now, cytoskeleton, the only thing to add is that it has two unique cytoskeleton components. We saw myofibrils in muscles, and cytoskeleton in nervous tissue in these cells have neurofilaments and neurofibrils. I just need you to recognize these two things as cytoskeleton components. That's all. Now with that, the last segment we have is that the axon is going to send signals away from the cell body, and then they are going to arrive at the endings of the axons called the axon terminals. And because dendrites receive, they are technically can be considered afferent, and because axons now, one thing to get about them is that, or understand about them, is that they send nerve impulses. So these receive and these send. And so you can see why I have axons away, dendrites receive. Or afferent. Efferent means to go away, efferent. These words are like the words superior and inferior. They mean into, and one means away. That's why you have afferent and efferent arterial when it comes to your urinary packet, which I don't seem to have open, so never mind. Oops, sorry. And so, in the perikaryon, that's where neurotransmitters are synthesized. Don't forget neurofilaments are a component of the cytoskeleton. And then we've made our way inside of the cell, and you can see nothing's different. Nucleus, a nucleolus. But one thing that's unique is the rough ER. The rough ER groups up into clusters and also with free ribosomes. Like here you can see free ribosomes, and here you can see ribosomes attached to the ER. These together are called nissl bodies because they make these unique structures. So these are clusters of rough endoplasmic reticulum, rough, and free ribosomes. And we know what ribosomes do. They produce proteins. And so, produce proteins. As a reminder, the nucleolus creates ribosomes, and ribosomes create proteins. Now, with this in mind, the axon has some components. When you look at the axon, it has the beginning of the axon. This is going to be called the axon hillock. And then the segment that follows that axon hillock is going to be the initial segment. And these two regions are going to serve as the gatekeeper for action potentials. And what this word is, action potential, it's nerve impulses. Nerve impulses, which is a combination of depolarization. And what was the other one? Repolarization. More on this later in the packet. But as I say nerve impulses, you are thinking simply like an impulse that's traveling this way, and then it communicates something to this cell, and this cell will in turn send it through something else. But when we say that an action potential has begun, typically we describe the first step, which is depolarization, and we're going to see that a little bit more. But you might see me say, instead of nerve impulses or action potential, you'll hear me say instead depolarize, which means the beginning of a nerve impulse. This is the second step of a nerve impulse, but more on that later. And so as we look at this here, initial segment, these are regulating that action potential. We'll talk about it again later. But this structure here, the plasma membrane of the axon, is going to be considered the axolemma, which means plasma membrane of the axon. And this little center space inside, this is going to be your axoplasm, which just means the cytoplasm within the axon. Nothing too difficult there. Now, action potentials only travel in one way. They only travel in one way. Remember this. Down here, you'll see that I have transport of materials. And when you think about an action potential depolarization event, I don't want you thinking about this topic, because I need you to see that this extension here is just more cell, right? Like more of the inside of this neuron. And inside of it exists a bunch of structures. Inside of it is a bunch of materials, such as ions, such as nutrients like carbs, lipids, proteins, lipids, like, lipids, fats. And so they're all inside of these spaces. Neurotransmitters might be flowing through here. And so these molecules can travel through the axon. That's referred to as axoplasmic transport. And so when materials travel along the axon, we call that axoplasmic transport. This can occur in both directions. When things are traveling towards the cell body, that's called retrograde flow. When movement is going back. In other words, it's going in this direction. Retrograde flow. That's what that means. And so to clarify, at this bottom note, you'll notice that I have down here, action potentials only go in one direction. Nerve impulses do not have retrograde flow. Action potentials and nerve impulses only go in one direction. But anything inside, anything that has to do with just cell-to-cell molecules, things that have to be made inside of the cell, materials that need to be moved through, they can go back and forth. But an action potential can only go in one direction. The last thing to add here is our telodendria. These are going to be, oh, there's an excess N here. Can you guys cross that out? Telodendria. And then, so telodendria are going to be branches of axon terminals, which are the ends of axons. And they're going to form connections with the next cell that it's connected to. This connection is called synapses, which is what this next page is on, synapse. And synapse means to connect, join, or unite. That's what a synapse is, where one cell, especially a neuron, primarily a neuron, will connect with another cell. And I want you to be cautious here. You'll notice that I wrote where neuron connects with another cell. I didn't say where one neuron connects with another neuron. I'm saying where one neuron connects to something else. Because if we look at this easy example, you can see one neuron connected to another neuron. Easy peasy. This one neuron connected to a skeletal muscle. You can see the thick and thin filaments on it. The sarcomeres. Oh, sorry. Here's the sarcomere range. You can see the little Z discs, the M line. And so these sarcomeres rely on your brain to send an impulse to communicate with it and cause the thick and thin filaments to interact together. It forces the skeletal muscle to release calcium from the terminal cisternae, and then the thick and thin filaments can contract. But before all that can occur, an action potential had to arrive from your brain and had to relay that signal. But this is an example of what we see one neuron with another type of cell. And this is that one that we learned in the last exam, neuromuscular junction. The muscle, nerve, and muscle connected together. And then lastly, we have neuroglandular. These are going to be neurons that are connected to glands. Your glands are under the control of your brain in the autonomic nervous system. And so they tell the glands when to secrete. And that's what this word means. Neuron connected to a gland. But you can see that the word synapse, it just means where they meet or connect. And so the cell that comes first is always going to be called presynaptic. The one that comes after is going to be called postsynaptic. And that's true for all of them. Presynaptic, postsynaptic. Presynaptic, postsynaptic. And so as we clarify the text and the image here, you can see presynaptic cell and postsynaptic cell. This means the one that comes before the connection. This is the one that comes after the connection. So if we were to zoom in on those axon terminals, we would see this object here. And we are seeing the axon terminal. We are seeing that it has a plasma membrane in contact with the plasma membrane of another cell. So we call this one the presynaptic membrane, nice and easy, because it's the cell that comes before, and the one that comes after is called postsynaptic membrane. Inside of these little axon terminals, all of them, right, as you see them, all of them possess inside of them synaptic vesicles that contain neurotransmitters. And neurotransmitters are chemical messengers and, like I said, hormones of the brain. And so when an action potential arrives, it induces these vesicles to release neurotransmitter into this space. And this should sound familiar to us because it's this very situation here. If we zoom in even more, it should look even more familiar if we look at this picture right here. So our action potential arrives. It induces these synaptic vesicles to release acetylcholine. Acetylcholine will be released into this space here called the synaptic cleft. These acetylcholine receptors will bind to the sodium channels. And when sodium begins to sweep through, what do we call it when sodium sweeps through the cell? Starts with a D. Depolarization. And so this is what depolarization means. It means to send us the first part of an action potential through the next cell, the post-synaptic cell, the cell that comes after. And so it relays the signal, and then to shut it off, these other little proteins had to come and grab these, break them apart, and make them useless. When they're like this, they cannot connect to this, and they cannot allow action potentials to go through the muscle cell anymore. And then it stops the muscle from contracting. And so all of this looks familiar because it's the exact same thing that happens wherever we see a connection with a muscle cell. So this picture as we see it now is the same thing, just a different illustration. And so this space is synaptic cleft. And that, as a reminder, is the space between the synapse. The last thing to add is that these axons can have a branch that extends off of them. We call that a collateral branch because they can be dynamic. They can hook up to another one of these neurons down here to its dendrite side. And so they have very extensive branching. And so when you see a branch off of the axon, collateral branch. Now, when it comes to neuron replacement, in most of the brain, you actually cannot repair cells. You can only slightly do it in your smell receptors, vision receptors, but I put here do not need. But the one that matters is that you can actually restore the neurons that are responsible for memory. You can have some sort of cell division with your memory, and working on things that challenge your memory have been shown to decrease things like dementia. So in our aging, we have to make sure that we're always working on our brain as we progress through life, the same way that you would worry about your joints and muscles when you're young enough to exercise them, that can lead to a more productive or a better quality of life in our older ages. And so learning throughout your lifetime can keep you sharp. And that's because learning is one of the only ways you can actually replace and create neuron connections, as a little fun fact. Now, these pieces here are the types of neurons you encounter. All of them are doing the same thing that we've been talking about, though. They all send action potentials, and keep it simple. Action potentials mean a nerve impulse. A nerve impulse or an action potential comes in two steps: depolarize and repolarize. We'll see that more further into the packet. But for now, as you look at these pieces, these extensions of these nerves, these are like the connections that you see on my iPad out here. You're seeing that I have an adapter. I have an HDMI, a power. I could connect it to this laptop with a USB. I can connect it with a mouse down here. That's what these things are here, what these neurons are doing, is that they will connect in unique ways, like the way that wiring and computer chips are in an electronic device. This is what your brain and spinal cord is primarily working with. It is using these in a unique connection to send signals in and out of your brain, to send motor commands and to receive or through your sensory commands, right? Sensory signals, things that are going into your brain. Now, anaxonic, we don't know much about it, but the word itself implies that it's missing or lacking an axon. And that's all I want you to know about it, missing an axon. Bipolar neurons are going to have two ends to them. And you can see that to the cell body, the cell body, you can see that it has two connections, one that leads to the cell body, one that goes away from the cell body, and you can see that they still have directions. Dendrites receive, but there's a difference. The dendrites are not on the cell body. They are on this little process, on this little extension, and that's what makes them different. This one is still called an axon. And typically you can tell when you look at axon terminals that the terminals have a more bulb-like shape at the very ends of it. You have these synaptic bulbs where there are connections, synapses. And these you find in the eyes. And also smell, olfaction. You'll see them a little bit later. The next one we have is our unipolar, and our last one, because technically multipolar is the same exact thing that we've been studying so far, the traditional looking one from the previous the previous pages. But this one, unipolar, this one's slightly different. How can you tell which one's which between bipolar and unipolar? The cell body in bipolar, you'll notice, is connected to these extensions. When you look at the cell body of unipolar, you'll notice that the cell body is completely out of the way. And we have two axons. And the only thing I want you to know about this is that typically it's going to be wherever you have, it's going to be mainly for your sense of touch, but again, senses. What are the other senses of touch? I'm going to expand on this a little bit. Whoops. Exactly right. So it technically only exists for those: touch, pressure, temperature. And then some of you might be like, okay, as annoyed as I was when I first had to kind of see that there's a distinction. But because, I mean, how do you tell the temperature of something without touching it? And I realized, like when I was cooking breakfast this morning and I wanted to see if the pan was hot so I can put my eggs in there, I didn't have to touch the pan and you knew how hot it was. So you can see how this is not touch technically. I didn't touch it to feel the temperature of the pan. So you can see why, little nuances, why in science is why they do this. But those are found mainly for the senses. And remember, senses mean those touch, pressure, pain, and the other one. So I'm just going to write senses. And I'm writing upside down. So emphasis, for senses. Not special senses. Although I will say I believe that they might exist in some of them, but mainly only in PNS. Mainly only in PNS. And lastly, multipolar neurons are the ones that we've been exploring, the first ones we started with. They're the most common in the CNS. Remember, CNS is brain and spinal cord. And then they are going to be the ones that are attached to most of your skeletal muscles to form neuromuscular junctions. And that's what my NMJ is there for. Neuromuscular junctions. Now, the majority of your brain, like I said, of your nervous system is made of these cells. In CNS and PNS, some combination of these cells. And they are involved in all communicating. But they can't do it on their own. They need assistive cells. They need their neuroglia. And so these neuroglia assist all classes of nervous cells. All classes of neurons, I'll say. In other words, these four cells, ependymal cells, microglia cells, astrocytes, the ones that we're going to learn next, they support these cells. They support these classes of neurons. And so these cells, to start, we start with our microglia. Microglia are something you already know what they are. They are monocytes and macrophages. They are white blood cells that engulf to destroy. Another word for this was phagocytosis. So they are phagocytic cells that engulf debris, waste products, and pathogens. Our astrocytes, you can see that they are linked to these capillaries, and these capillaries are going to contain plasma. And remember that capillaries are exchange sites. These astrocytes, you can see that the capillary is completely guarded by these astrocytes with their little leg extensions. And the job of these astrocyte cells is to transport materials from the capillaries, like let's say it's useful water or oxygen, and then that oxygen can be transported, and then your nerve cell right here, you can see that this is an axon it's connected to. It can put that oxygen molecule, can put those useful ions right into the neuron's axon if it wanted to. And so that's what astrocytes do. You can see that they're regulating what's going through the blood vessel and going into the nervous tissue, and they can even remove stuff from out here and then place it inside and either remove it out here or send it out. As you look at it, they are the cleaners and regulators of what goes in and out of the cells and their extracellular fluid, the ground substance, the interstitial fluid. They regulate it. And so these astrocytes, because they do this very important role, they're called, or they create something we call the blood-brain barrier, which means that they isolate the CNS from blood. And with that comes, what does the capillaries, what does plasma have that it needs to be regulating? Easy. All the things that plasma has: ions, nutrients, gases. And so those things are regulated closely by the astrocytes, and they don't just let anything in, they only let the things that your brain needs, because they know that if they let in the wrong things, it can kill the brain. So they only allow the essentials in. Now, oligodendrocytes are going to be involved in creating our white matter. And actually, before I get into this one, let's do the ependymal cells. The ependymal cells, these cells are found in the ventricles, which I believe most of you, if not this week, you'll be learning it in lab if you've done brain yet. But if you haven't, when you do brain, you will see that there are these chambers inside of the brain that look like this. And you don't have to flip pages here. This is easy enough and labeled for you already that it's easy enough to understand. That even though we haven't fully gone into the entire brain just yet, we know that there's some general concept around that there's this large mass of the brain. And you might have seen in your notes when you've sort of been going through your lab packets that you're seeing sort of a ram's head inside of the brain. And this circulates as you look at it. It's hollow space. You can be swimming in there. If we could magically pop up in there as little tiny little human beings that just are in here, we'd be swimming in here in a fluid called cerebral spinal fluid. In all of the blue space that you see inside of that brain. You can see it looks like a ram's head. This is from the side. And you can see it makes its way all the way down, not shown, but it goes all the way down the central canal, which is part of the spinal cord. And so inside of these chambers, you find ependymal cells. And so as we go back to your packet, you have this page way further ahead, but those are what your ventricles are. And so these cells create cerebral spinal fluid for you in the ventricles and the central canal of the spinal cord. One last time, as we look at this image, you have CSF flowing through those blue spaces. It circulates throughout. The ependymal cells are lining in there, and they create this cerebral spinal fluid. And then that makes its way all the way to your central canal of your spinal cord. And so that's why you can see in your photo that they put up here a little spinal cord. This is a little spinal cord cut out so that you can see it. If you were looking at it like if you chopped down a tree. And you can see that there's this little circle here. That's what your central canal is, and your entire spinal cord has it. And you can see that you can have the word central canal right here because it's showing you that inside of that central canal, you also have ependymal cells. And ependymal cells do what? They produce CSF. And so they place it into that central canal and it circulates with the ventricles. Again, it's circulating in that central canal, made maybe a little bit in there. They're made in the central canal and made in the ventricles. So as you look at this one last time, ependymal cells are in here, in the ventricles. This is the terms that you have in lab that are under ventricles. And when you get there, and I'll cover this now so we don't have to do this page when we get there, you just have to know the same things that you know for lab, know the same exact terms for lab, be able to label the ventricles inside of the brain. And don't forget, connected to the central canal of spinal cord. Yours is on, I don't believe these. Yeah, you're going to have to go through the brain packet a little bit. Kind of in the middle of your packet. So that's the relevance right now of that structure. Circulate CSF, connected with the spinal cord because of that central canal. Ependymal cells are responsible for creating CSF, which is cerebral spinal fluid. Oops, sorry, wrong way. Wrong way. And so we go back to that page. Now, we move on to oligodendrocytes. Now, oligodendrocytes help the neurons by providing a framework. In other words, they kind of hold it up together. And when we look at myelin, myelin is the name of this lipid-like material, this very sticky material. But what it's meant to do is that it's meant to speed up nerve impulses. And so it does so by creating a myelin sheath. I don't know if any of you know what a sheath is, but if you watch a samurai movie, you know where the samurais have their sword and they take it out, out of that little thing that their sword container. That's what a sheath is. And so this is sort of the same thing. Because if you look at our photo, you have a neuron cell body, they have axons that go through, and they are surrounded by myelin, this fatty material. And you can see that the axon, if you make a cut of it, it's sitting right in the middle of that myelin sheath. And because of this myelin, right, you myelinated axons, it's saying that these axons have myelin. You're going to be soon learning throughout your medical careers what demyelinating disorders are. Those are disorders where this myelin sheath are wearing out on the axons or being destroyed by your own body sometimes. And when that's happening, your brain, your consciousness, your thinking begins to deteriorate. Because without this myelin, you don't conduct action potentials or nervous impulses adequately. And so these myelin sheaths here will speed up nerve impulses. Now, the only thing to add about this now is that as you look at these structures, you'll notice that the oligodendrocytes, that they are cells themselves that place their little leg extensions and they are wrapping around different axons at the same time. So you can see why they're considered that they're therefore framework, because it's like they're holding up many neurons together. They're sort of gluing different axons from different neurons together. And so you can see framework. They're holding it all together. They're even connected to your astrocytes. Your astrocytes will feed them the nutrients that they need in order to make that myelin sheath and keep it healthy. And so you can see why we consider them part of framework. The only last thing to add here is that you'll see the word white matter and gray matter when talking about the brain and spinal cord, the CNS. And when you look at this word, white matter and gray matter, it only means because it's only called that because the white matter is where you have a bunch of myelin. But I want you to think about this in a second. It's going to matter. You see gray matter. This is where you don't have myelin. And what does myelin what's the function of myelin? Good. Speeds up action potentials. And action potentials travel only through what? What part of neurons? Axons. And so as you look at the image that we looked at before, and I'll just go ahead and pull it up. You don't have to pull it up yourselves. Whoops, sorry. That when you look at this, you're seeing gray matter and you're seeing white matter. So if you thought about the structures that are here, what do all these dots represent then? And what do all these little extensions represent? The green would be axons, and then all in the gray matter, it's only cell bodies. Does that make sense? That's how the brain is organized. As you look at it, white matter only contains axons, and all the cell bodies are in the gray matter areas. Because you don't need myelin on cell bodies, you need it on axons. So as you look at the organization of how the brain is organized, whenever you see these two tints of color, white and what's supposed to be gray, what you're really seeing is the millions of cell bodies you have in the gray matter. And in the white matter, it's mainly going to be bundles of axons with myelin on them. And so even the spinal cord itself looks like that as well. The cerebellum, you can see gray matter, all the cell bodies, and all the axons that belong to those cell bodies are completely grouped and bundled together, and they're all heading out to deliver the nerve impulses that they need to deliver. Or they're receiving them, we don't know. But I hope that makes sense simply, that as you look at this, you learned a lot. One of the most complicated things at an advanced level, if you can recognize that gray matter means there's a bunch of cell bodies, there's a bunch of these parts of the cell, all grouped up in the gray matter, and then white matter is pretty much all the axons bundled together. So even if you were to grab the spine and chop a person in half and look right at the spinal cord, this is what you would see. You would see gray matter and you would see white matter out here. These are all neuron cell bodies, and all their axons that belong to them that are myelinated are going to be out here on these white matter portions. And I hope that's clear.
So it's the opposite on the spinal cord?
It's the same in the spinal cord. Gray matter is always cell bodies.
No, but like the positioning.
Oh, yeah, kind of. Yes. No, I will say kind of. Kind of, yes and no, because even the brain has centers to it that are gray, really gray. And I don't have a photo of that to show you, but a good observation in the brain, yes. For the cerebrum, yes. But there are centers in the middle of the brain that have gray matter, mostly gray matter. But yes, good observation there. Now with that. Now more of the same. Oligodendrocytes produce myelin. This is so important that we sort of have to recognize that oligodendrocytes are only producing myelin in the CNS. And so you'll notice that on the next page I have myelinated in the CNS is done by oligodendrocytes. And then you'll see that the leftover thing to do is that we have to discuss PNS. PNS, the cells that myelinate those axons are not called oligodendrocytes. Pardon my writing here, but it's Schwann cells and neurolemocytes as you see them right here. Schwann cells and neurolemocytes. This neuroglia, Schwann cells, exist only in the PNS. And where it's slightly different, the oligodendrocyte, it used to use its little extension, like its little leg extensions, to make the myelin sheath. But with a Schwann cell, the entire cell is going to wrap itself around to create a myelin sheath part, right? Here's a Schwann cell, a Schwann cell, a Schwann cell. So you can see there's multiple Schwann cells creating the myelin sheath. And that's the big difference. Oligodendrocytes are like little octopus using their legs to create huge myelin sheaths. And in the PNS, the Schwann cells use their whole bodies, their whole little cell bodies, to wrap around the axon. And they don't even cover the whole thing. They only cover a little piece. So they need many more little Schwann cells to come and help them out. Big difference, right? And you can see that axon running right in between. And like a sheath, just like in the other instance, it contains inside of it an axon. And Schwann cells are their own little cells with their own little nuclei, just like oligodendrocytes are. So the only thing to add here, make sure you're aware of, they do the same job, but oligodendrocytes in CNS, Schwann cells in PNS. Satellite cells do not worry about, that's why I have a little DNN there. It might probably come up a little bit later. Myelinated sheaths on axons give the appearance of white matter. Many myelinated axons. The only thing, last thing that about myelin is that it creates unique little regions. For example, here in the myelin sheath, you have gaps in between myelin sheaths, like this, like this, like this one. These are called nodes. And the space in between them is called internodes, between the gaps. They used to be called nodes of Rambier, but just know them as nodes and internodes. Really easy. Keep it simple. And don't forget that where you do not have myelination, you will have the appearance of gray in either the brain or spinal cord. So the CNS gray matter, the gray matter inside of that central nervous system, brain and spinal cord, is mostly going to be cell bodies or unmyelinated axons, right? There's no myelin on those axons. Lack of myelin makes them appear gray. In other words, you have more of this in gray matter. The only last thing to add, nodes and internodes exist in Schwann cells as well, but it works the same way. Gaps and space between the gaps, internode. You'll notice that each one of these Schwann cells is a cell, and it's called the neurolemocyte. And then just like the word axolema was the plasma membrane of the axon, this neurolemocyte has a plasma membrane, and it's called a neurolema. And so that is the plasma membrane of the Schwann cell. And you have to make that distinction, because if I said, oh, the plasma membrane at the axon, you don't know if I'm talking about this plasma membrane of the axon or the plasma membrane of the neurolemocyte of the Schwann cell. So that's why we have to create those words. And this is an example of how it forms that sheath. Many layers wrapping around it to make that sheath. And so we keep going. We do not worry about Wallerian degeneration. I'm going to remove this out. And then I want you to go straight to the brain structures that look like this on your packet, because I sort of remodeled it a little bit. And I know that there's the action potential stuff, but I want to record that in a special one. So we're just going to go ahead and go straight to this picture right here, because this will help you with lab this week if you're all in brain, which I would imagine you are. And then the second part of this video will contain the action potential part at the very end. Once you know all these structures, learning action potentials is perfect at the end. And I just kind of wished in hindsight I would have moved it to the end. All right. So for the last remaining part of this first part of the recording, we're going to finish these general structures. And they are the same ones in lab, so a lot of this will help you with lab. So the brain itself, that model that you've been seeing on your bench, looks like this, right? Looks like a layer, a group of like foldy, moldy, cottage cheese texture looking thing. You can divide that whole thing into 4 regions, and you should see it in 4 regions. You have your cerebrum. Oh, they're right here for you. Cerebrum, cerebellum, diencephalon, and brainstem. Starting with the cerebrum, we're going to approach each one slowly and simply. The cerebrum is going to be the outer region that you see around the brain. It has these internal structures. Whoa. How does that keep happening? And then see these internal structures within the brain. And you can see they remove it out. And so we're only focusing on everything on the outside right now. This section is called cerebrum. This cerebrum contains landmarks such as small grooves we call sulci. We should be familiar with the word sulcus, which means it's singular for groove. Sulci is plural for small grooves, but many grooves. So sulcus, small groove, sulci, many small grooves. I guess we can add that. And so the brain has a bunch of small little grooves inside of it. And then you have larger grooves we call fissures. And the example of this is going to be this fissure that runs right across here that separates this lobe down here from the one up here. And this would be an example of a fissure. Another more apparent example, you don't have to flip your pages, I'll do it for you. But you'll see that if you look at the brain, it has 2 halves, right? 2 hemispheres. And these 2 hemispheres have a longitudinal fissure that separates it here. So that's the difference. A sulcus is a small groove. It ends right there. And a fissure is a very deep groove. And so that's the difference between sulcus and fissure. The last word you have on there on your page is gyrus. That's going to be the surface, not the grooves. And so we finish up that little segment with gyrus is the name of the flat surfaces that are in between the grooves. In other words, all of these flatter regions, gyrus. Gyrus. Gyrus is singular, gyri is plural. And it's pronounced with a J, gyri. Next little lobe to talk about here is our cerebellum. Our cerebellum contains some grooves and some sulci, but we don't have to know any in our section. What I need you to know about the cerebellum is that its main function is going to be in balance, coordinating motor commands from the cerebral cortex. And so what that means is that it regulates balance. And we'll talk about this word right now. Now, when we see these 2 words, and I like to start off here because you'll notice cerebrum and cerebellum, easy to confuse. And the hardest thing about nervous is not understanding what it does, is not confusing the language. So when you see the word cerebrum, and we're referring to anything within the pink areas, the cerebrum, we call it cerebral. Cerebral, well, it's an ugly A, cerebral. And cerebral implies being within the cerebrum. If we say cerebellar, it implies being within the cerebellum. And so you'll see down here we have the word cerebral cortex. Cerebral means in the cerebrum. And remember, the cortex in Latin means area. Area, specifically outer area mostly, right? And so your cerebral cortex is where you are going to be carrying out conscious thought, like motor commands. And actually, it's already there. I don't know why I'm writing it. Motor commands right here. You're also going to use that area to store things that you've learned. You're going to make sense of it, process it. And then you also receive inputs from your senses and special senses. And now this diencephalon. This diencephalon contains two structures that matter, thalamus and hypothalamus, or matter for our class. And the thalamus and hypothalamus, what they serve is that the thalamus serves as a sensory relay. That means that when you see things, the first place they go to is your thalamus. You hear things, the first thing that those signals go to your thalamus. You feel a sense of touch, it goes up your spinal cord from your skin receptors and then goes to your thalamus. And then your thalamus decides, okay, this is sight, this is hearing, this is touch, this needs to go to, sight needs to go to this part of the lobe, touch needs to go to this part of the lobe, hearing needs to go to this part of the lobe. So that's what I mean by relay center. That everything that's going in an afferent direction, going into your brain, will go to your thalamus first, and then that thalamus will send it to the next area. More on that later. Hypothalamus, has to do a lot with endocrine. It's typically referred to as your master gland because it is involved in a majority of your hormone production. It also does a majority of your autonomic functions. Again, an example of this was going to be smooth muscle. Smooth muscle contractions, cardiac muscle, glands, what's part of your autonomic functions. And lastly, for emotions. Remember, autonomic are things that we do not want to control. Easiest ones to think about are the muscular layer of all our digestive tract, those smooth muscles that are in there, and cardiac muscle, the cardiac muscle within the myocardium. And then we have our brainstem, which contains the midbrain, pons, and medulla oblongata. The midbrain is going to be an area where we contain visual and auditory reflexes. And what this means is that, let's say you're walking in the dark, and actually, let me get rid of all this extra. When you're looking at this region here, when we say visual reflexes and auditory reflexes, this means that if you're in the dark and then all of a sudden you're walking, you're going to the bathroom right in the middle of the night, and then you hear a noise like a little pitter patter, like of a rat or something, your cat, your dog, or something moves, what can you do, right? You like, whoa, like your head goes naturally to the sound. That's what auditory reflex is. Or right now, if I were to grab my phone right now and just chuck it at one of you right now, what would you do? You would like, whoa, you would like dodge it, or you would put your hands up. That's visual reflexes. Your midbrain handles that. And so that's where visual and auditory reflexes are, in the midbrain. This is where you also find a structure in lab that they're going to ask you. It's going to be called corpora quadrigemina, and it's going to contain the superior plus inferior colliculi. I don't ask you this in here, but I know it's in your lecture. And that's going to be these two objects. I think I printed out the labeled one for you. Does that one have it labeled? No? Okay. And then so
Not that part.
this is what that whole summary is. This midbrain in green has areas responsible for visual reflex and auditory reflex. The superior colliculus is the top little balls on top. There's two. I'm going to put two so that when you get to lab, you'll know what I'm talking about. Ask your professor if you're not sure. And I don't ask you in here, but for lab, it's worth mentioning. And altogether, because there's two on top, two on the bottom, corpora, like cuerpo, means body, of four, and that's what it means. And then so you can see it's a body of four parts. And so 1, 2, 3, 4, you have one on each side. Superior colliculus, inferior colliculus is singular. You might hear this called colliculi for plural. Lastly, we have our pons, and our pons, its job is to connect the cerebellum to the brainstem. Connects cerebellum to the brainstem. And this is going to be involved in motor control. Both skeletal, I'm going to put both voluntary and involuntary. And the last piece of that brainstem is your medulla oblongata. This is going to be where you connect our spinal cord to the rest of the brainstem. But mainly what I want you to know it for is that in that region, you also have very important autonomic functions such as heart rate, breathing rate, that are found in those areas. You'll hear them in physiology called pneumotropic, things like that. Little regions that control things like breathing rate and heart rate. But for us, autonomic functions is all I need you to know. And so the lobes are the easy part here. When you look at the lobes, they're named after the structures or a part of the bones that we know or we learned in the skeletal system. Frontal lobe, parietal lobe, temporal lobe, occipital lobe. And there is one new one called the insula. And they are all part of the cerebrum. Only the regions that are found on the outside. And so flip to your next pages here. This is a better photo for this. And you'll see that they're color-coded for you. And so the only last complicated thing to add here is that each lobe is going to be responsible for your senses and your special senses. For example, we start here at number 6. Number 6 is where your visual area is, right? That's what the word cortex is. This is the only confusing part about this is the language. In this occipital lobe receives all visual stimulus, meaning everything you're seeing on this presentation screen, you are receiving it and processing it in your occipital lobe right now. And so where you receive the signal, right, you receive the visual, then you see that you receive it. And then you get it from that information from the eyes. This is accurate, but this is more complex. But we're simplifying this. You're going to eventually receive it from the eyes, and then they connect to this. But we don't care about this beyond the scope of this class. So the eyes send impulses to that occipital lobe eventually. The visual association area in this occipital lobe, you have this part that doesn't receive, but it processes, it interprets what you're seeing. What's the difference between receiving and processing? If you're looking at on a presentation board, like right now, you're seeing the letters C-A-T. You are seeing, that's it, just letters C-A-T. What does it mean to interpret it? What happens when you put this word together? Cat, right? And what's a cat supposed to be? Like a little animal a pet, right? Or a wild cat that you see. But this in and of itself, what in the world does this have to do with a cat, this symbol? What in the world does this symbol have to do with a cat? Or this one? So you see how you see this, but you interpret cat. You see patterns in it, and that's what your occipital lobe does. So the only thing to really address in this whole page is primary visual cortex means the area that receives the first visual, primary visual. The association area is the part that makes sense of it. Okay, what color am I seeing? Is it blue? Is it sky blue, cyan blue, dark blue? I don't know my colors that well, but you get my point. Is that you can tell the differences, or you can interpret what you're seeing in that same lobe, the occipital lobe. And it gets just as easy, it's just as easy here. Auditory cortex, your temporal lobe. It's going to be the area where you receive sound, and then you interpret it. You can interpret different sounds like the letter A, and O, right? You can determine the difference because in your temporal lobe, you not only receive signals, you also interpret them. And so though that temporal lobe contains the primary auditory cortex and the auditory association area.
Professor,
Ask away.
what if your occipital lobe, what if you're blind? Does that heighten other senses of the other lobes?
It's possible that some senses are better defined when you are blind, for example, like smell and hearing. And all I'll say is that damage to the occipital lobe can cause problems to vision. We know what the brain does only by trauma and injury, because when certain parts are injured in accidents or disease, we see it affect certain parts of your abilities. And so in your question to this context, if you were to damage the occipital lobe, it would affect your ability to not only see, but maybe also to interpret what you're seeing. You might not be able to see where one thing ends, like as you look at this door here, this might look blurry, the exit. It might look blurry if you're looking at it and you had some damage in your occipital lobe. You can't see where one thing ends and one thing begins. Or you might have trouble with color. You might forget certain things that you've learned visually. So occipital lobe, just an area responsible for vision. Temporal for listening, auditory. Next easy ones here, gustatory, gustos, no me gusta, right? This means taste, your ability to taste things. Your insular lobe receives information of taste. Olfactory is smell. Your insula regulates smell as well. So all impulses of taste and smell go to the insular lobe, which is found in between the brain, in between the temporal and frontal. You kind of have to move them. That's what they're showing you. Insula. Now, to finish up the last two senses that we have only senses really, right? We did our special senses. Now, sense of touch is handled by parietal. And this is going to be, again, sense. We call it the area that contains primary somatosensory cortex. This means your senses. Again, that means touch, temperature, pain. That's right here for you. Touch, pressure, pain, vibration, temperature. Sense, not special senses. And then you interpret those senses. The only unique thing to add is that the parietal lobe has one unique region, which is actually the area that receives all of those senses. That's going to be the post-central gyrus. And the rest of it is going to be an area where you interpret it, your association area. And that means to distinguish between different sensations. If I put a penny and a dime in your pocket and I tell you you can't look inside, but you can figure out, you have to give me the penny, You would know how to do that just by sense of touch. That's what we mean by association area. And then the last thing, this central sulcus divides that region from the frontal lobe. The frontal lobe handles all conscious control. It is where your personality is stored, your frontal lobe, and it's also where you contain your motor cortex. And think about it, your personality, who you are, what you're going to do, whatever it is you are, at the end of the day describes, your personality is defined by what you do every single day, right? Who you are. And so you carry out everything you've ever desired or wanted. You do so with muscles. You move around to do so. And so this is where you control all your actions on your day-to-day lives. And much like this other half in your parietal lobe, the frontal lobe has this segment called precentral gyrus, which is where you send out all motor commands and control all your skeletal muscles. This region before is an association area. This is where you put all the things that you've learned, like in sports, like dancing, writing, anything that requires learned motion. You store it in the premotor cortex, and that makes up your frontal lobe. And so to summarize, your precentral and postcentral gyrus control different things. This one's found in your frontal lobe. Controls your primary motor cortex, voluntary movement. When it comes to touch, your senses, that's handled in the parietal lobe. Put in parietal. And that handles body touch.